GDF11 levels, there's been age-related dysfunctions in mouse skeletal muscles. And the starting point of the research is beginning in these skeletal muscles. Obviously, the muscles are, tissue comprised, and there are multi-ligament fibers. So we're using these muscles to actually analyze the progressive stages in aged mouse and unaged mouse and their effects within. Okay. So this next slide is about they come in when the muscle is injured or is aging, I guess you can say. The normal cell that you see, the satellite sits outside the cell and it really doesn't do anything. But when it's starting to regenerate, it becomes activated, it goes into the cell, and it actually proliferates and differentiates to become, I guess you can say, more myoclonus. So it compensates for the injury that's actually occurring. This, just to keep in mind, is the old mice and the young mice and what happens between them. The myofiber is in the muscle of the young mice, and compared to the myofiber in the older mice, the myogenic cells are basically hold a space and compensate for the space of myofibers in this.
We're going to continue.
How the actual hold is on decline. They're actually holding on to the function that they do. So, yes, it's apparent.
Up in here.
This is basically the method. In the paper, they depict it as, obviously the two mice, aged and unaged. They don't specifically tell you how the blood from young to old go into the subjective mice, but they do mention that there are incisions made in each of the mice. Then they sew them up by first elbow and knee to help with the mobility and skin. And after the incision, they do phocytometry to actually kind of the differentiation of the blood between the two. Yes. So it actually goes in and out from both of the mice, which is very interesting. And there's no actual vein or like blood combining sites, should I say. But they put them side by side and they align the subjective mice, and they're focusing on just their central region area between the aged and the unaged, and how they correlate with each other. And they're actually observing the properties between the aged mice and the unaged mice.
Of their blood. So we
are basically concluding that aged mice have less foreign colonies than satellite cells, which basically means that there is more DNA damage than in aged mice than unaged mice. So there isn't too many regenerative properties necessarily regenerating through the mice. And as you see the age to young comparison subjected, there's not too much comparison apparent between them. So that's that. Graph B is actually evidence of DNA damage, images of the cells that so, and the most damage that became apparent throughout the research. This up top is the consent cells of where it's aged at and the amount of damage that it definitely does have. So the question is, so the figure C shows that this is true. I'll go through the top. Daphne is a scanner for satellite cells up in the top, and it shows to the grooves of the DNA. It's apparent all throughout in young as well as age. And UAX is a and it shows basically when DNA is damaged and the histones are actually phosphorylated around it. So this is the marker for phosphorylation. And when you see the green, it's basically saying that there are phosphorylated histones, which implies that there's actually DNA damage. So you can barely see anything in the young ISO or with the young mice in general. And as you work your way down, it actually becomes less apparent in the aged head. And that is really something that was focusing on because we're trying to get regenerative properties either from the young to the old or from the old to the young. So the aged head obviously means that young to age, with the age being apparent, is actually some type of regenerative properties going on throughout this process. The merged category obviously is for all of the parent, and there isn't any DNA damaged in the head. So it's very apparent that it actually is concise between all three, young ISO, young het, and aged ISO. Yes. Alrighty. So we're going to continue on. Basically a quantitative graph, or basically the figure that you see the cells obviously on our y-axis, and we have them range from the young iso, young het, and aged iso. And as you can see, leading factor would be aged mouse. So with that, we find that it was the aged iso mouse, excuse me. So with that, we found that there were actually the regenerative cells, and it actually shows the parent in the data that this is what we were actually looking for. So, answer to the question, is blood from young mice able to reduce damage in aged mice? Yes. And we see that just because the evidence that's been shows, because of the graphs that have been apparent throughout the entire slide. So I do think that there should still be some continuation of experimental study to just create certain factors of reassurance on the research that has been involved. I do believe that my hypothesis supported the data that is shown. Thank you. And my references are at the end.